What we have learned in these 6 months from the beginning of the outbreak, the first outbreak has been huge. And as you mentioned before, we have tons to learn about the virus, but all the community have been moving forward, and that has been possible because collaborations among academia, agencies, stakeholders, producers who have been willing to test and participate in research.
I would just echo what Zelmar said. I think this has been really a demonstration of how important it is for us to have these existing foundational relationships across the industry and across academia and different disciplines, because it doesn't just affect one part of the industry. This is affecting the entire industry. And I think that it really is important for us to make sure that we are keeping those connections strong and moving forward.
Hello and welcome to this special episode of Dairy Digressions, the new podcast of the American Dairy Science Association, the Journal of Dairy Science, and JDS Communications. I'm Matt Lucy, former editor-in-chief of the Journal of Dairy Science and former editor-in-chief of JDS Communications and current physiology editor for JDS Communications. Dairy Digressions is where you get an inside look at the science and the scientists that are making headlines both inside and outside the dairy industry. We explore the cutting-edge research that creates knowledge and drives innovation in production and processing of milk for human consumption. Hey everyone, help me grow this podcast. All you need to do is like and subscribe, and of course, listen and spread the word to a friend or colleague. Now let's get to today's special episode. Today, we highlight a special hot topic issue from JDS Communications entitled Highly Pathogenic Avian Influenza in Dairy Cattle. This special issue has two editorial comments, one from Jess McCart of Cornell University and a second from Kay Russo, who's a consultant in Colorado, and three hot topic articles. We have two of those hot topic authors with us today, Dr. Nicole Martin from Cornell Food Science and Dr. Zelmar Rodriguez from the Large Animal Clinical Sciences at Michigan State University. Welcome, Nicole and Zelmar.
Thank you.
Hello.
Fantastic to have you guys with us today. We're talking about H5N1. The American Dairy Science Association, along with JDS Communications, has sponsored this special hot topic issue. And what we are trying to do is get information out as quickly as possible. So this hot topic issue on H5N1, it is open access, which means that you, as a member of the dairy science community or outside the dairy science community, can click on the issue and read everything that is there. This special issue is one in a series of special issues that we've published. The latest one was on dairy cow behavior. Again, these special issues, as well as the entire content of JDS Communications and the Journal of Dairy Science, comes to you without a paywall, free of charge, because these are open access journals. Today, we're going to talk to these two authors, but I just want to review the fact that this special issue contains two editorial comments, one by Jess McCart, in which he discusses the need for collaboration at this time across the dairy industry during the time in which we have this virus out there. And a second editorial by Kay Russo, who's a dairy consultant in Colorado, where she just basically discusses the need for action now, not to sort of fool around with this any longer. And we make sure that we work hard on this right now. And then there's Nicole's article, which has to do with on the food science part of the equation, Zelmar's article, which is basically epidemiology. And then finally, there's a third very nice article from Salman Butt, who is from Cornell, which is a very nice article, which discusses a little bit about the host range of this virus and how it infects so many different types of species. So it's definitely worth a look, again, October 11th. We're going to start with Zelmar, and we're going to put you on the spot right away, okay? And we're sorry to do that, sir. Dr. Zelmar Rodriguez is a dairy veterinarian and assistant professor in the College of Veterinary Medicine at Michigan State University. He worked in the dairy industry, completing a PhD. She's a DVM, completed a PhD as well in veterinary epidemiology from the University of Minnesota. And he has an integrated research and extension program. And you're mainly interested in milk quality, antibiotic use, and disease control. So this is a perfect topic for you. The topic of your article is epidemiological and clinical aspects of the highly pathogenic avian influenza H5N1 in dairy cattle. So the first question I'll ask you, Zelmar, is Can you tell me what word I'm supposed to use when I describe what we're talking about? Should I say HPAI H5N1, bird flu, all of the above? What is the most correct term to talk about this?
I prefer to refer technically as highly pathogenic avian influenza H5N1. It's a bit long to be repetitive on the term, so
Yeah.
H5N1 would be more appropriate.
So my understanding of this is that it started in Texas.
Yes, influenza we know is not new. H5N1 have been in the US in poultry in 2021 in December, but just recently in March of 2024 is when we identified the virus in cattle. So there was an event of bird to cattle transmission. What is fascinating about this is after that, the virus started to spread cattle to cattle and eventually to other small mammals and humans.
So this is quote unquote bird flu. And I know it's devastating when it comes to poultry. I mean, whole flocks just gone. Was there some sort of bird that had a mutation and then a mutation allowed it to hop to dairy cows? Or was it the exact same genetic sequence that was killing birds that just ended up in a cow randomly? Or was there a mutation? That's the question.
There has been there because it's not exactly the same virus itself, but it's very, very similar. So when the samples were collected in clinical animals in Texas, and those correlate very, very similar to those in birds. That's why we know that moved from birds to cattle.
So it's a slightly different virus from the one that was originally found in birds. I mean, it's the same virus, but there's a mutation there. Is this new one that we're dealing with in cattle, is it more likely to infect a human, or is it about the same as all the other previous viruses to infect humans? Because I know some of the concern, not only for dairy cow health, is that it's going to infect cattle, but are humans at a greater risk at this point in time than they were previously?
Each clade is different in that regard of virulence and ability to move between different hosts. But the challenge and the big concern that we have now is that it moved from cattle to humans. We don't have any evidence so far that it moves from human to human, but eventually if we have positive herd, the virus is circulating among cows. There are many workers working in close proximity with these animals, in which density is sometimes very large, especially in big farms. And they are susceptible to seasonal flu. And if they get infected additionally with this HPAI cattle virus, then there may be some reassortment and potentially transmission from human to humans. And that is why it's very important, not just from a dairy production perspective, but from a human point of view, that we try to mitigate and reduce as possible the spread of the virus in dairy farms, within and between.
So as I understand it, the cows present initially with maybe some respiratory symptoms, but ultimately what they really notice is the effect on the milk from the cow. Can you explain what would be a clinical presentation of an infected cow so our audience sort of understands that?
Well, actually, that's one of the problems of this disease. The symptoms are generally vague, so there are some mild respiratory signs, there are GI signs, in some cases there are diarrhea or tachyphysis, there are some fever, which is difficult to measure sometimes, but there are fever, there are anorexia, drop in rumination, but all this leads to the drop in productivity, and that is what is more evident for a producer, from a farm worker. It's very noticeable, the drop in productivity, the increase in somatic cell count driven by the drop in productivity and also by the neutrophil migration into the mammary gland. Mm-hmm. So I would say, if we think on the abnormal milk, there is a change in coloration, there is this colostrum-like milk aspect. Also, at the population level, when we see these cases of abnormal milk that you test them on farm or in a lab and they came negative, and you have many cases at the same time, and that is totally different that we are used to see on mastitis type of diseases or other diseases. So that is what is very striking and is the driver of us suspecting of the virus in the herd.
Just to summarize what you said, when we have mastitis in herds, it's typically a small number of animals might contract on any one day. But when you see a large number of animals with very similar symptoms, like you're stating, a big drop in milk production and sort of funny color to the milk, that wouldn't be a normal kind of situation with mastitis. So you're like, whoa, there's something going through here, like some sort of pathogen is moving through this herd. Clinically, what is a veterinarian going to do, prescribe when they see this thing moving through the herd? I've heard, just for example, that get all the affected cows on fluids or something like that, or put maybe salts in the water so they really drink a lot of water. Is that just me misinterpreting? I'm not a veterinarian. What are you going to do? You walk onto a herd, you're like, whoa, we've got H5N1 going through here. What are you going to do as a veterinarian?
First of all, we have to be clear that this is not a bacterial pathogen, so it's not that we're going to use antibiotics.
Oh, yeah. Okay.
Right? So that's the first thing. It may serve for secondary infections, but not for the virus. What we can do is mitigate the symptoms, so make the cow feel better. I've been talking with some veterinarians that are using aspirin to reduce the fever, fluid therapy. So those approach help for sure, because if the cow feel better, drink more water, then drop in productivity is not as high if we don't do anything at all. But the only thing that we can do is mitigate the symptoms and make the cows feel better.
So I guess it's a little bit like when we get the flu, they say, oh, drink lots of fluids and you have a virus. So unless you're going to take the antiviral thing, you need to move through it and let your body deal with that. So keep the cows hydrated and so on.
Yeah, essentially it's the same with humans, right? We have seen that the symptoms tend to last for 5 to 7 days in general. Most cows recover productivity. The fever lasts for a very few days. Yeah, that's what we have to do, just helping the cows to move forward. It's easy to say, right? If you have a large herd, you may have dozens or hundreds of cows during the peak of the outbreak with signs of disease. And it's really, really challenging to work with all those cows. And sometimes they require multiple days treatments. So it's a big burden for workers and producers for sure.
Would they move those cows to the sick cow pen and separate them from the herd? Or are they likely to try to manage them with the rest of the cows in the herd?
I've seen both. Actually, I've seen hospital pens overcrowded, and at some point during the peak of the outbreak, you don't have space for more cows. Some producers have kept the cows with the rest of the herd. We don't have an enough sample size yet to make an inference here, but we have seen in this herd that the clinical cases are still commingled with the rest of the apparently healthy cows. The incidence of the disease larger as compared with those that segregate cows.
If it was possible, it might be best to move them to a sick cow pen, or at least get them away from the main herd if possible.
Yes.
And once a herd has this, is it a one and done? All my cows are better now. We're all good. Is it a one and done, or is it type of thing where it just goes on and on and on?
You know, we don't know yet, at least as much as I know. We have seen that milk production after, usually the outbreak lasts approximately 2 months in general.
Oh, wow. Ooh.
Between 1.5, a bit more than 2 months, but it's around that time. I've seen in a couple of herds a recovery and then new drop in productivity, but in those herds, still the bull tank test came negative. So we cannot say that it's a reinfection of the animals. We still are trying to figure out how long the antibody lasts, the response lasts. That will be a determinant if we're gonna have multiple outbreaks within the same herd, or at least we will have the herd protected for, let's say, the whole year, right? We don't know if it will become a seasonal event or not yet.
Do you think, now I'm jumping ahead a little bit and we're going to get back to the testing, is this going to be the type of thing like what we do in people? Every fall we get our flu vaccine and we're good to go? Or is it just, I'm on planet Mars when I talk about every fall cows will get their flu vaccine and we're good to go? Is it that sort of a virus, or is that not the future of this thing?
Well, personally, I think that came to stay. In humans, influenza is more seasonal because the weather, the temperature. But in cows, we have a system in which cows are stressed. They deliver, they start production all the time. You always have cows during the transition period. So they are immunologically challenged all the time, right? And if you have the virus circulating in the environment, it may happen that new naive animals get reinfected or infected. So I think it's going to be here for a very long time circulating. The situation, like the big picture is still cases are coming up every day. There is no reduction in cases. On the opposite, there are more and more cases, so the virus is spreading. So yes, I think we need to be prepared for that.
But there's no commercial vaccine at this point.
Not at this point, no. That is more a political decision, I think.
Political? Why is it a political decision?
Well, because there are certain trade regulations related to vaccination of animals that may play a role in there. But I think that eventually we may hear more about it.
So I was reading your paper and you published a graph, figure 1A, looks like weekly cumulative cases in the United States, and it gets up to like 200 herds or something like that. 200. That's got to be a vast underestimation of the reality, isn't it?
Yeah, it's very likely to be an underestimation. When this paper was accepted, 2-3 days later, we hear about California became a positive state to the virus. And in a span of a week, pretty much, they got 40 farms with infection. It's not that, I don't think that spread in 1 week to 40 herds. I think that it was a political decision, was the willingness to producers to test. And that's why we saw what we saw in California. And now we have in Colorado, for example, they have mandatory testing. And we saw that after the mandatory testing started, they got an increase in cases until they reached sort of a plateau. I would expect something similar happening with the other states too.
So Nicole, I think I'm going to lean on you for a few minutes, if you don't mind. And I enjoyed your paper as well. Dr. Nicole Martin is an assistant research professor in dairy foods microbiology and associate director of the Milk Quality Improvement Program in the Department of Food Science at Cornell. This is where I just stalled. Zelmar, you got to know how these people are in New York, okay? Nicole grew up in the beautiful southern tier of New York State. I am from like central New York, okay? And these southern tier people just think that they have the most beautiful part of the state there is.
It's lovely here.
So, where she began, she worked on a local dairy farm, and she has BS, MS, and PhD degrees from Cornell. And who was your mentor at Cornell?
I had a couple. So Catherine Bohr, who was the dean of the College of Ag and Life Sciences for quite some time and is now the dean of the graduate school. And then my PhD mentor was Martin Wiedemann, who I now work still closely with here in the Department of Food Science.
That's quite a good pedigree. Congratulations.
Only the best.
Only the best.
It's because I grew up in the southern tier of New York. I don't really know what to tell you.
The Finger Lakes people are going to argue about this, okay? I mean, I was just skinny.
Listen, I'm now a Finger Lakes person, so
that's true. You are. Zelmar, do you know what we're arguing about? Have you ever been to New York?
No, not much.
Please schedule a trip and I'll show you around.
Ithaca is gorgeous, as they say, right? In your article, you brought up 4 important aspects to understand about this. And you brought up, first of all, the initial viral load. And can you just explain what sort of numbers you're looking at or thinking about?
Yeah, I think that was one of the big shockers with the development of this disease, is just how much virus is being shed into the milk. The estimates range pretty broadly, but it's not uncommon to see 10 to the 8, right? Which is just exceptional, extraordinary numbers. And as a food scientist and as someone who studies food safety, I know that the equations of sort of foodborne illness include what is that initial concentration of the pathogen of interest, right? And when we have those very, very high concentrations to begin with, that's obviously a concern from a food safety perspective, because the virus is not going to replicate in the milk after production. So we're not going to get any higher. But when we start out with those exceptionally high numbers to begin with, that's obviously a big factor that we're looking at from a food safety perspective.
So you could have a single cow that somehow could get milk that's going to be a big number of virus particles coming out of that cow.
Sure. And obviously there's a lot of factors here about what goes into that bulk tank or what gets processed. But we have small herds. We have small herds that, sort of bottle their own milk or make their own dairy products. And then we have situations where there are a lot of herds going into the milk supply of a single processor. And so we start to think about what's sort of the distribution of the concentration of this virus going into our milk supply. And certainly it's going to vary, but the fact that we have such high shedding, such high viral loads coming from these infected animals is a big issue.
Now, I see you have a minor in microbiology. Okay, so here we go.
A minor in microbiology and a minor in animal science.
I'm going to think back to my virology. So just so our listeners understand that there's a biological reason why this virus ends up in the mammary gland. There are receptors for the virus in the mammary gland. And if it's like the other viruses I know about, the virus will infect that cell, replicate inside that cell, and then I guess kill the cell and be released into the milk. So it's replicating inside the mammary gland. So that's why we get its really high viral load within the mammary gland.
I'm not a virologist either, but I am very fortunate to work with some very wonderful virologists here at Cornell. And the tropism, as they say, for the mammary gland, this is where this virus is found in dairy cows, and they can find it in other locations in the animal, but not in the same concentrations, right? So in the sort of respiratory tract where you would, I guess, expect to find an influenza virus, it's found in much lower levels. So it is very much a mammary gland virus.
Again, so people are like confused. This is supposed to be bird flu and if it was just giving the cows influenza, then we wouldn't worry about the milk because maybe it never would get into the milk. But in this case, for whatever reason, this guy is infecting the mammary gland, and therefore this is where a lot of viral particles are being shed. I think one of the reasons you're into this and so deeply is the raw milk question. Okay. And so just to kind of cut to the chase, I think what you're going to tell us is that the typical process, pasteurization will basically inactivate this virus. I think you're probably going to tell us that. So for those that don't know, milk is homogenized, and that's mixing it up to deal with the fat particles, but it's pasteurized, and that's to kill pathogens. Okay. But then there's this raw milk thing, and this raw milk laws differ state by state.
Yeah, so the bottom line is that pasteurization is very effective at eliminating this virus. And there have been a number of studies that have come out over the last few months. Some of the initial ones, they weren't quite as illustrative, but there have been some very thorough and robust studies at this point that would demonstrate that pasteurization, as we use it here in the United States, is effective at eliminating this virus. But we do have raw milk consumption in this country. And in fact, we have increasing levels of raw milk consumption. And that is potentially a risk for consumers who are choosing to buy, purchase, and consume raw milk, because we have this ongoing outbreak where we have potentially very high levels of virus in milk, and then consumers who are choosing to consume that product. And this research is ongoing. We don't know everything yet. We know that high temperature, short time pasteurization is effective. We know that pasteurization is effective, but there are a lot of other processing mechanisms that we use in the industry that we are still trying to gain clarity on what the impact of those techniques is on infectious virus.
Just like, for example, that milk that goes into cottage cheese or sour cream, is that pasteurized first?
That's right. And then typically receives a much higher heat treatment as well. Depending on what the product is. We like creamy yogurt. We like creamy sour cream, and heating it to very high levels for a long period of time is what actually denatures the proteins and allows us to have that creamy mouthfeel. So those products are receiving much higher heat load than your typical gallon of milk that you pull off of the grocery store shelf. Again, the sort of baseline there is pasteurization, and that pasteurization is very effective.
You mentioned there's pasteurization and the UHT, which is ultra high temperature. That will also inactivate the virus.
Absolutely.
Has there been a single case of someone drinking milk or eating a dairy product in America getting this virus?
There have been no reported cases of that route of transmission. We actually know from studies that the FDA did that the pasteurized milk supply and also other products, not just fluid milk, but butter and cream and other ice cream products, that they tested for remnants of the virus, right? An activated virus using a PCR test, that the sort of virus particles were found in the dairy supply, but none of them were still infectious. So that's more evidence that the commercial products that are out there for sale are safe for consumption. There are products that are treated at sub-pasteurization temperatures, and those are somewhat concerning. We have some new research coming out of Cornell here in a little bit that we're looking at what are called thermization processes. Those are sub-pasteurization heat treatment processes. And for the most part, those are very effective as well, although those can take on a lot of different time and temperature combinations because they're not legal pasteurization. So processors do their own thing. And mostly that's a cheese issue, right? For raw milk cheeses, which are made frequently. The risk that we see as from a food safety perspective is really from a raw milk consumption and a raw dairy product consumption perspective. Although, again, we don't have any cases that we know of consumption leading to infection of humans. And really, it's a theoretical, sort of transmission pathway. Anyway, although we do know that animal models in research and also animals who have consumed the infected milk from the infected dairy cows have contracted H5N1 and come sick and in some cases passed away.
I think it was mentioned somewhere in here about people would feed waste milk to cats and stuff, and the cats would become sick. Okay. And that's what you're talking about. Waste milk on a dairy is milk that's not going into the bulk tank. Sometimes people feed it to their cats because they love their cats on dairies. It goes to this day. And so that would be evidence that if you consume high dose of this virus, it could potentially be harmful. I want to Nicole, can you just explain to our listeners so it's very clear the difference between a positive DNA test and an infectious virus particle, because some people are going to hear that on the radio. Oh, I found H5N1 and it's on my DNA test. Can you just be very clear so people understand?
Certainly. So we can test for this virus in a couple of different ways. The fastest and easiest is to do what we call polymerase chain reaction or PCR test. And that is simply a test that looks for the RNA of the virus, so the genetic material of the virus. And finding that genetic material does not mean it is an active or infectious virus. It means that it was there at some point, but it could be inactivated, for example, through pasteurization at that point. In order to determine if there's an infectious virus, we have to go another step into more complicated, either cell culture experiments or other methods that are used to determine if that virus is still active. To be very clear, you can find the genetic material without there being any infectious virus.
Right. It's not a living thing. It's a dead thing. Right. So for you guys that love woolly mammoths, who doesn't love the woolly mammoth? Just because I can sequence DNA from a woolly mammoth doesn't mean I have a living woolly mammoth. Okay. And that's the same thing. Just because you can sequence the DNA or the RNA from one of these viruses doesn't mean it's a living virus. And that's important, So just remember the woolly mammoths. We're not there yet. Just so our listeners understand this, dairy farms produce milk. They collect the milk into bulk tanks. All bulk tanks are sampled. Every load is sampled. I might be telling it wrong, but Nicole, I think every load is sampled because we have the samples, because we have to test for antibiotics in milk. So every single load of milk has a sample with it, and it goes to the processing facility. So we can test for this virus, but we don't test for this virus. So for example, Zelmar, theoretically, the state of Michigan could say, okay, Zelmar, here you go. Here's all the samples from the last month. They're coded. You won't know what farms they're coming from, but we're going to give them to you so you can at least tell us how much is out there. But you're not even at that level, are you? There's no sharing of these milk samples to test. I guess it would be illegal. You'd have to do that at night in a dark alley or something. But what are your thoughts on testing? I mean, you mentioned in your paper, I thought it was really interesting. You said two important aspects of epidemiology of HPAI H5N1 is that we don't really know how herds initially become infected, and we don't know the dynamics of transmissions. If you don't know what herds have got it or when they got it, it's almost impossible to understand what we're dealing with. I understand that dairymen, they're worried that something bad will happen if their herd shows up positive, like the processor will say, well, you got a positive tank here, we're dumping all your milk. Is that a possibility, Nicole? Like if a tanker came back and it was positive, would they potentially just dump your milk?
I think that's the concern, right? Is what do you do with that product then? Because processors also don't want to assume the risk of viral particles, let alone infectious virus being identified in their product, right? So it does have those downstream consequences. And I certainly think that we would be in a situation if we were testing at that level where those tanks wouldn't be accepted.
It's an interesting question, isn't it? There's 30,000 herds in America. I don't know what capacity we have for testing, but it would be a heavy lift to test all herds in America, every tanker from every herd in America. At this point, we really don't probably know how much is out there. Nicole, do we know how many herds are out there in New York State?
The last census was around 2,700 farms in New York State, which is obviously way down from what we were a few years ago, but still a lot of dairy farms.
Mm-hmm. Do we have a sense for how many might be positive for H5N1?
At this point, none that we know of. But as you've been discussing, there's a barrier to testing, even under voluntary conditions where we're trying to just get a sense of where this virus is. It's not something that a lot of folks are eager to do.
It gets tremendously complicated, tremendously fast. If you started testing, and this was a lot more widespread than we ever thought. And again, I think what I'm trying to say here is that we can have herds that might be asymptomatic that may come back with some viral particles. And I'll just explain to our listeners that the tests we use to test for these things are extremely sensitive. And if processor says, I won't take it, I have a zero tolerance. This gets really interesting. If I have zero tolerance for viral particles in this tanker, it'd be interesting to know what percentage the U.S. milk supply would be impacted. It's an interesting question, isn't it? That we don't know the answer to. Zelmar, you agree with that or no?
Yeah, we don't have an answer for sure.
Yeah. And I don't know what the answer to that is either. I know that because we don't really know how many herds are carrying some level of viral particles. And, we know that if you take a look at antibiotics in milk, we know that our antibiotic testing in milk has been insanely successful in eliminating. I mean, antibiotics just don't end up on the grocery store shelf, in part because we're testing, but in part because we just don't get that many positive bulk tanks. Is that fair enough, Nicole, to say that we don't see that many positive bulk tanks for antibiotics?
Absolutely. It's very rare. And I think that that's one of the core tenets of the Pasteurized Milk Ordinance, right, is withholding milk from sick animals or treated animals. Mm-hmm. And I think that at this point, that's just something that, we know it's not happening. It's not possible to withhold milk from cows that are infected with H5N1 completely because they're shedding virus well before they're symptomatic. And Zelmar might have a better figure for us at this point of how many days prior to clinical symptoms they're actually shedding the virus, but long enough that this milk is getting into the milk supply, right? It's there, and we're not withholding, even if the intention is to withhold.
It's the subclinical stuff. It's the stuff where they're shedding virus, but they don't have any symptoms. And we deal with, of course, subclinical mastitis cases, and we deal with it all the time, and we know how to manage that. We don't really know how to manage this. Is that right or wrong? I don't really know if I'm saying the right words because they're shedding virus, but we don't know they're shedding virus.
Yeah, I think it's important to mention that you may have, for sure, subclinical cause to the virus, but you don't have subclinical herds. So if you have animals shedding in the herd, it's expected that it will be catched by the bulk tank test, right?
Right. So those cows definitely, and I think, Nicole, what you said is there are regulations that say if a cow has abnormal milk, there are regulations that say you should not be putting her milk in with the other cows. Is this correct, Nicole? And
that's right.
Pasteurized milk?
Pasteurized milk ordinance.
It just basically says if you have a cow with abnormal milk, it doesn't go into the bulk tank.
That's right. So from a safety perspective, from a food safety perspective, which the Pasteurized Milk Ordinance is designed to protect the safety of the milk supply, that is one of the most important components is to remove that milk from the supply.
I don't know if it's right. I always tell my students that milk is 10% of the human diet. I don't know if that's true, Nicole. You probably know, but all right. So I'll just go with it. Okay. And then I read it somewhere. It's 10% of the human diet. Okay. And the other thing I think that's important about milk is it's a very special product because it's considered to be healthy and clean. And we also know that the cleaner the milk is, the longer its shelf life is, and the better the flavor the milk has. And so this pasteurized milk ordinance, it's not just a law to make people's lives difficult. It's a law to ensure that we have clean milk that's good tasting and a healthy product to consumers. And speaking to that ordinance, if you look at things like somatic cell count, which is an indicator how clean milk is, and all the indicators of how clean milk is over the past 100 years.
It's 100 years this year, actually, Matt. This year is the 100th anniversary of the Pasteurized Milk Ordinance. Yes.
And we can say very specifically that we have cleaner milk today than we've ever had before. Is that true?
We have lots of data that would indicate that in the measures that we use for milk quality at the farm, that milk quality has improved consistently over the last 100 years, and somatic cell count being one of those, and bacteria counts being the other. But, the reality is that before the Pasteurized Milk Ordinance, 25% of food and waterborne diseases were due to consuming milk that was unsafe. And now that number is less than 1%. So we have very clear indication that this regulation does have an impact on public health.
Yeah, so I think it's important that any consumer listening to this understand that this ordinance is in place. I don't think we have dairy farmers knowingly, with cows that are infected with this, knowingly putting milking units on this cow. It doesn't make any sense. Any sensible dairy farmer would realize, I don't want to put a milking unit on this cow. She's probably going to spread it to all my other cows, and I don't want all my other cows to get this either, And I think it's fair to say there's a real commitment among American dairy in general to make sure they sell healthy product. We not only sell healthy product, but we also do some things in the processing side to ensure it stays healthy. But I want to get back to Zelmar and just ask the question. Okay, so we talked about vaccines. You say there's no vaccines ready. Would it be of value for dairies to just lock down their herds? In other words, close their herds completely to outside animals? Would that stop the transmission of the disease if they said, okay, this is a closed herd? Would that stop the transmission of disease, or is that not going to really do it?
Unfortunately, probably not. We don't have very clear answers on how the disease transmits from herd to herd. We know that cattle movement is a major transmission route. We still don't know the role of air transmission in not just within the herd, nose-to-nose contact, but also the particles flying long distances. We are speculating about the role of fomites, about people carrying the virus in their boots, in their clothes. We know, for example, that in poultry, carrying the virus even in the clothing is devastating for flocks. We don't know if that's the case in dairy herds. It's likely that people play a role also, and we know that when we talk about a closed herd, we are talking about not bringing cows, buying cows from outside. But what about people? Many farms share personnel with other farms. They have some part-time workers, or those workers live in the same house that workers in another farm. So it's absolutely difficult to actually fully close a herd. I think it's impossible. You need haulers coming to your herd and taking the milk. So all the vehicles, there are for sure biosecurity measures that we can implement to minimize the risk of introducing the virus. But at the end of the day, having a closed herd is not a bulletproof for sure.
I think what I'm taking away from this conversation is we're very early in this H5N1 situation, and we need research and knowledge to really understand how we're going to go forward. That's the role of science, right? As scientists and agricultural scientists, we faced a lot of challenges, but I would argue that particularly in the dairy science community, we have some of the strongest researchers and the strongest scientists in across animal agriculture, and as well as agriculture in general. I'm just going to put it out there, agriculture in general. You don't have the stability of the United States food supply without a lot of really solid scientists behind it. This is just another example of where a large number of solid scientists are going to come together and figure this thing out. But I get the sense when I talk to Zelmar and I talk to Nicole, these two are experts. I get the sense that we are in very early days here in terms of understanding this and understanding whether it's a one and done, it'll be gone next year, we'll never see it again or whatever. I think there's lots of work to do, lots of late nights ahead. Well, Zelmar, you're an assistant professor, so it's good. You need research topics for the rest of your life, right? I'll just put in a little shout out for a few things. It sounds like there will be some abstracts at the annual meeting of the American Dairy Science Association.
I would expect to see some, yes.
Yes, I would see probably from both your groups. And I think it's going to be fun. We're going to have lots of great sessions on H5N1. And the American Dairy Science Association is in Louisville this year. It's June 22nd to 25th in Louisville, Kentucky. And the abstracts are going to open very soon, Nicole. In October, the abstract portal is going to open, but I think they close much later. But you can at least get your abstract in.
February, yes.
In February, they're going to close. The other thing we have is a webinar coming up. Are you both participating in the webinar?
Yes.
ADSA webinar, sort of mid-October.
October 16th.
October 16th.
Yes.
I think you have to register or something, October 16th, okay? And these guys are going to be a webinar, and they are going to dive deeper into this topic. But you guys are super busy, and you guys have just been fantastic. I'm going to allow you to, if there's anything that you felt like we did not cover or you would like to just emphasize, is there anything else? I'll start with Zelmar. Anything else you'd like to sort of bring forward before we close today? Anything else?
I mentioned that what we have learned in these 6 months from the beginning of the outbreak, the first outbreak has been huge. And as you mentioned before, we have tons to learn about the virus, but all the community have been moving forward. And that has been possible because collaborations among academia, agencies, stakeholders, producers who have been willing to test and participate in research. And I think it's very important to mention that thanks to all those stakeholders, we have been able to know what we know, and we are going for more.
That's so important about the agricultural community, isn't it? That we have tremendous support from, producers, the universities, industry partners as well, who really care about the United States food supply. This is just going to be one of those things where everybody works together, because you can't, Zelmar's not going to solve it from his office at Michigan, and Nicole, she's from Cornell, so they're going to solve it all. And that's kidding, Nicole. But Nicole, do you have anything else you would like to add?
I would just echo what Zelmar said. I think this has been really a demonstration of how important it is for us to have these existing foundational relationships across the industry and across academia in different disciplines, because it doesn't just affect one part of the industry. This is affecting the entire industry, and I think that it really is important for us to make sure that we are keeping those connections strong and moving forward. We don't know what the next thing that's going to come along is, but this has really opened our eyes to the fact that we kind of, need to keep our eyes open for new things and what's next, and just keep those foundational connections strong.
Thank you very much. And I think one of the things listeners, I think you get a sense that these two people care, and they care about the people they serve. I just feel like I knew when we did this podcast, you two would be a success because you two care. And I appreciate your time very much. So we're going to conclude with a few words, but thank you very much for your willingness to participate. I do appreciate it very much.
Absolutely. Happy to do it.
Thank you, Matt.
All right, everybody, that brings us to the end of our show. If you enjoyed Dairy Digressions, be sure to like, subscribe, and rate us, and spread the word to a friend or colleague. If you have feedback for us, please let us know at adsa@adsa.org. For show notes and more episodes of Dairy Digressions and learn about everything, go to the ADSA@adsa.org site. Until next time, thank you everyone for listening.